In this lesson, I'll talk about cancer. This lesson includes control of cell division by mitosis, role of mutations, tumor formation, types of tumors, and metastasis. So let's see control of cell division by genes. Control of cell division by genes. If this is, for instance, an animal cell, this is a normal cell. It has chromosomes present in its nucleus. If this is, for instance, a pair of chromosomes, it undergoes G1 phase, during this phase, protein synthesis takes place. Cell decides to divide or not to divide. If this cell has decided not to divide, it is rested in a quiescence phase, which is known as G0. But if it decides to undergo cell division, it grows in size, passes through G1 phase, and synthesizes proteins. So if this cell completes G1 phase, it enters the S phase. During this S phase, DNA is replicated. So the chromosomes are duplicated in this case. So each chromosome now will have 2 chromatids. Before S phase, each chromosome has only 1 chromatid. So there are 4 chromatids at the end of S phase or in the beginning of G2 phase. So replication of DNA that takes place in an S phase. At the end of G2 phase, during this G2 phase, the cell repairs the damaged DNA. So repair of the damaged DNA takes place. Duplication of centrioles takes place. So the cell is prepared during this interphase to go into an M phase that undergoes division. So this is an interphase. Now at the end of G2 phase, the cell will be ready to undergo M phase. It will have the same number of chromosomes and the same number of chromatids as at the beginning of the G2 phase. Now during this M phase, which is mitosis, the nucleus of this cell is divided into two, and finally at the end of this cytokinesis, this cell is divided into the two cells, two daughter cells, each with the two chromosomes. This is the normal cycle of a cell. If there are all possible factors available and the proteins synthesized normally, the cell will go through this G1 phase into the S phase. If DNA is replicated, then it goes through the G2 to an M phase. And during an M phase, the spindle fibers are formed and one cell is divided into the two cells. This is controlled, this continuous progression of one cell to go through the different phases and finally divide into two cells is controlled by the genes. There are two types of genes that control this cell cycle. One are the proto-oncogenes. If this is, for instance, a normal cell, it is rested in G0 phase. If it is not dividing, if it has committed not to divide. If it has to undergo division, it passes through this cell cycle, which I have reviewed already, and this is controlled by the genes. There are two types of genes found in this normal cell. One are the proto-oncogenes and others are suppressor genes. So these are suppressor genes and proto-oncogenes or activator genes. Proto-oncogenes control the synthesis of proteins which are known as inactivated proteins. activator proteins. These proteins control the progression of a cell through the different phases to finally undergo the cell cycle and divide into the two cells. So, if these proteins are present in higher concentration, this cell would undergo rapid division and will make more and more cells. If these proteins are lower in concentration, more the proteins, more are these proteins, higher the rate of cell division, and vice versa. However, suppressor genes control the synthesis of proteins which are known as suppressor proteins, and these proteins inactivate the activator proteins. Inactivate the activator proteins, or inhibit the activator proteins. So they stop the cell from undergoing through this cell cycle and slow down the mitosis or stop the cell from dividing. These proteins may bring a cell to repair the damaged DNA or bring the cell to undergo apoptosis. So if there is everything normally provided, there are the normal growth factors present there are enough proteins synthesized, the duplication of the cell organelles has taken place, this cell would undergo this cell cycle normally and will continue to divide one cell into two cells. However, if there are any mutations in either of these genes, maybe the mutations, if there are mutations in this proto-oncogenes which changes it into the oncogenes. These genes become activated and they increase the release of these proteins. Increase the release of these proteins. So as a consequence, the rate of cell division that increases. So cell continues to divide without stopping at any phase. Similarly, if there are any mutations in these suppressor genes, these proteins are not synthesized. If there is a mutation here, this protein is not synthesized, and so the activated proteins are not inhibited. The cell does not undergo the repair of the damaged DNA, and it does not also die by an apoptosis. So consequently, this cell continues through or progresses through this cell cycle and continues to divide into the cells without stopping at any phase. So that is how mutations in any of these two types of genes, either the proto-oncogenes or the suppressor genes, that lead to an unstoppable and uncontrolled division of cells by mitosis, which leads to the formation of a mass of undifferentiated cells, which is known as a tumor. So let's see, how do we define a cancer? It is an unstoppable division of cells by mitosis. This unstoppable division of cells by mitosis takes place due to mutations in genes which control cell cycle, result in an uncontrolled cell division. Mutation is a random process. It's a random process. However, there are some factors which increase the chance of mutations. Such factors are known as mutagens. Every mutation does not lead to cancer. And also, every mutation is not inherited to the offsprings. Mutations in specific genes like proto-oncogenes and tumor suppressor genes may cause cancer. So, the factors which cause cancer are known as carcinogens. Some carcinogenic substances include carcinogens, include radiations, chemicals. There are some viruses which can also cause some types of cancer. Now let's see the development of cancer or the formation of the cancer. As I discussed earlier, that the normal cells, if these are the normal cells, they can enter quiescence or G0 phase, and they stop dividing and they do not undergo further cell divisions. However, if mutation in a cell takes place in the proto-oncogenes, which are transformed into the oncogenes, or in the tumor suppressor genes, so this fail to stop dividing and continues division. Generally, when cells are damaged in an organ or tissue, like when you get an injury on your skin, so the surrounding cells, they send cell signals the Malpighian cells or the stem cells in the skin, they respond to these cell signals, and they stop dividing or they continue dividing depending upon the cell signals. But this mutated cell will fail to respond to the chemical signals. It fails to respond to the chemical signals and it continues to divide. So it produces a mass of undifferentiated cells. To prevent the proliferation or the division of the cells that have damaged DNA or that have mutated DNA, there are immune cells or immune responses in our body that remove these cells with the mutations or mutated cells by the T lymphocytes. In many cases, these tumor cells or these mutated cells are removed by these lymphocytes from our body. So they do not develop into the cancers or do not develop into the tumors. However, these cells sometimes fail to be removed by the immune system and continue to divide by mitosis and increase in number. They produce an undifferentiated mass of cells. These cells are different from the surrounding cells and they all look alike. They all continue to divide and produce more and more cells. So rapidly, this increases in mass and looks different from the surrounding cells under a light microscope. These dividing cells have a higher need of nutrients which if these nutrients are not provided, so this may have a limited growth. But over a course of time, the blood vessels may develop inside this tumor. There may be the capillary network that develops in this tumor, and it continues to provide the nutrients to the dividing cells. So the dividing cells continue to increase the mass of this tumor. This tumor is known as a benign tumor. This may remain limited to where it is first formed and is known as primary tumor or primary growth. So this is primary growth. Sometimes the cells of this primary tumor are removed and carried to the other places in the body through the blood vessels or through the lymphatic system. And these cells establish themselves in various other organs or parts of the body and start a new tumor, which is known as a secondary tumor or secondary growth. This is secondary growth. This process of spread of these cells from a localized tumor to the other parts of the body is known as metastasis. And when this tumor is metastasized, such a tumor is known as malignant tumor. If this tumor become metastasized, then it is known as malignant tumor. This malignancy causes tumors to spread from a localized area to the various other parts of the body, and it then becomes difficult to control the spread of the tumor. That's it about cancer for today.